Hey everybody, my name is Valerie Marcano, and today I will be presenting necrotizing hepatitis associated with Clostridium perfringens and Broiler Chicks based on samples that were submitted to the Poultry Diagnostic Research Center and the Georgia Poultry Laboratory Network. 12 submissions of either swabs or tissue were evaluated. Of these, 11 were submitted between January and April 2017 to either the PDRC or the GPLN, and one was submitted in August of 2019. All birds were between 1 and 10 days of age at the time of submission. 10 pertained to one line of breeders, and the other 2 submissions belonged to a separate line of breeders. They were across 3 companies located in 3 different states, 10 flocks residing in Georgia, 1 in Indiana, and 1 in. Elevated 3-day mortality was indicated in 10 out of 12 flocks. An elevated 7-day mortality of up to 5% was indicated in all flocks. Neurological signs were also observed in 10 out of 12 flocks, and they included tremors, stargazing, and incoordination that started at 7 days of age. Gross lesions observed in the field include liver lesions, which were areas of tan and or green to dark brown discoloration on the livers, as can be observed in this picture. In 10 out of 12 flocks, congested lungs were also observed, as well as serosanguinous fluid in the caudo-ventral abdomen and gas bubbles in the yolk sac. Next, we will look at histology and stains of the different lesions that were observed. But first, because of the damage observed in all samples submitted, let's have a brief reminder of what a normal liver looks like. In this picture, there's a portal triad containing the bile duct, the hepatic artery, and the portal vein. As we zoom in, we can see a 2-cell layer thick hepatic cord with bile canaliculi in the middle. Mild to severe and multifocal to coalescent necrotizing hepatitis was observed in all 10 liver submissions. There are areas of acute coagulative necrosis, which are pointed out by the asterisk, and these are mixed with large numbers of rod-shaped bacteria, which on a gram stain were revealed to be gram-positive, and these you can see pointed out by the arrow. Lesions were mostly perivascular and periportal, and often associated with vascular thrombosis, infarcts, and a large number of rod-shaped bacteria, as can be observed by both the arrows and the asterisk. Here is a close-up of one of those areas of hepatic necrosis with elongated rods, and on the second picture you can see immunohistochemistry done for Clostridium perfringens, which is positive as denoted by the dark brown color. In addition, here's another example of a Clostridium perfringens positive immunohistochemistry. These are within the vessels and bile duct, as pointed out with the arrow. 6 submissions also contained sections of intestine. Of these, 2 had mild intestinal congestion and mild hemorrhagic enteritis. 6 of those, on the other hand, had necrotic enteritis with myriad of intralesional bacteria that were also consistent with Clostridium. This is a section of small intestine which had severe necrotic enteritis, and an immunohistochemistry was done for Clostridium perfringens and was positive. We also had one sample of brain that was submitted, and in this submission, vacuolization of the white matter parenchyma was observed, or cerebellar spongiosis. These likely led to the neurological signs that were observed in some of these birds. We had one sample of heart that was submitted, and in this case we found a fibrinous pericarditis. We also had one sample of spleen submitted, and fibrinous splenitis was observed in this case. This slide summarizes our results. Submissions 1 and 2 were hatchery samples, so they included egg yolk, meconium, hatchery damper swabs, eggshell swabs, basket fluff, and coil coolers. Of these, all except for the coil cooler samples were positive for Clostridium perfringens, and it was isolated in culture. Submissions 3 to 12 were tissue sample and necrotizing hepatitis due to Clostridium perfringens perfringens infection was observed in the submission of all 10 of those flocks. All of these flocks were also still younger, 10 days of age or younger. Clostridium perfringens was cultured in 5 of these submissions. Immunohistochemistry for CP was positive in 7 flocks, and of these, necrotic enteritis was only confirmed in 4 cases, but again, only 6 submissions of small intestine were made. In total, 11 out of these 12 flocks were positive through bacterial isolation or immunohistochemistry. It is worth noting that one of the Clostridium perfringens cultured was positive for NetB. This is one of the isolates only. It was the only one tested. And in addition to being positive for NetB, it was positive for the TPEL gene. TPEL is known to enhance the virulence of NetB positive Clostridium isolates. Follow-up was done with all 3 companies. However, we only heard back from one of the companies. In this company, environmental samples and egg touches were done at the hatchery. So these were our submissions 1 and 2. And then gastrointestinal tracts were submitted for both aerobic and anaerobic bacterial culture and profiling. NetB positive Clostridium perfringens was isolated in broilers, pullets, and breeders and had a 14% prevalence. Of these, broilers across complexes ranging between 21 and 31 days of age had a 6 to 14% prevalence of NetB positive CP. In addition, random amplification of polymorphic DNA or rapid fingerprint showed similarities between isolates among these broilers, pullets, and breeders. Specifically, identical fingerprint was seen in one isolate that was from a 1-day-old chick, and it matched an isolate from a broiler breeder. This makes us wonder about vertical transmission. Now let's take a step back and discuss generally Clostridium perfringens. It is often associated with necrotic enteritis with or without cholangiohepatitis in turkeys and chickens older than 2 weeks of age. Coccidial challenge is thought to be a predisposing factor. However, the presence of maternal antibodies can also help delay the onset of disease. Clostridium are spore-forming, gram-positive aerobic rod-shaped bacteria. They replicate in the intestine of poultry, and CP specifically is enterotoxin producing, having NetB as the main virulence factor. NetB belongs to the alpha-hemolysin family of beta-pore-forming toxins, which means that it damages the membrane of the cell, and a lot of the damage is due to osmotic changes. In addition, TPEL is associated with NetB, is present in about 10% of the isolates, and it can enhance the disease severity caused by NetB producing Clostridium perfringens. Other enterotoxins produced include CPE and CPB2, which have also been suggested to play a role in necrotic enteritis. Disease can be subclinical, with only seeing reduced weight gains, decreased feed conversion, and increased condemnations at the plant due to cholangiohepatitis. Disease can also be clinical, including sudden onset of high mortality, dehydration, depression, ataxia, water-filled crops, diarrhea, anorexia, and ruffled feathers. Transmission can be horizontal or vertical. There was one report from Shane et al. From 1984 in which Clostridium perfringens was isolated from the yolk sac of one embryonated egg. Presumptive diagnosis can be done through clinical size and gross lesions, but before confirmation, histopathology, bacteriology, PCR, or immunohistochemistry should be done. During necropsy, you may see distended and friable, thin-walled jejunum or ileum. You may have petechial hemorrhages, or you may have the traditional Turkish towel, which is a dipteric membrane covering of the mucosa of the small intestine. In addition, the gallbladder may also be distended with bile and may have necrotic foci on the wall. Swelling and congestion of the liver may be seen with multifocal pale yellow to greenish hepatic necrotic foci. For control, antimicrobials such as BMD, streptomycin, lincomycin, penicillin, and oxytet are used. In terms of prevention, because coccidia is a predisposing factor, coccidia control is very important. In addition, antibiotics, probiotics, organic acids, and vaccination can be used. In this case, antibiotics such as BMD or avelonicin can be efficient. For the company we were able to follow up, broiler breeder testing was done. Probiotics were used, and for breeder flocks that were positive for Clostridium perfringens, penicillin was used as an antimicrobial therapy, in addition to sanitation both at the hatchery and at the farm, including nests, belts, buggies, holding rooms, etc. After treatment, both 3 and 7 day mortality drastically went down, as well as neurologic signs. Concluding thoughts. This is the second report of necrotic hepatitis in chickens 10 days of age or younger. This included one report in 2003 by Sasaki et al. Newly hatched chicks may be infected. Again, the article from Shane et al. From 1984 demonstrated the presence of Clostridium perfringens in the yolk sac of one bird. In addition, they were able to isolate it from the intestine of a chick that was younger than 7 days of age. This prevalence of Clostridium perfringens in commercial broilers and hatcheries must be evaluated, as we are isolating it from eggshells and chick fluff, from chick papers. Maternal antibodies may also be important, as they are present during the 2 weeks of age. All of this information points at the importance of parental flocks and hatchery as critical control points for Clostridium perfringens. Thank you all for listening to my talk. I would like to thank Dr. Monique Franza and the rest of the PDRC, Drs. Gamble and Mashek, and the rest of Pilgrim's Pride Corporation, Dr. Anna Villegas Campbell, Dr. Oscar Fletcher, Dr. Jim Davis, and Dr. Bridget Fruin. Thank you all very much. And if you have questions, please come to the Q&A session. Thank you.